So today we are going to discuss about the concentration cells. So let us just put a heading for that firstly. So we are to discuss about the concentration cells. So far we have been seeing that the potential in the galvanic cell, it was being produced when the different types of the electrodes were present in the cathodic and the anodic compartment. That means in the one case we were taking the metal ion half cell, and in the other half we were taking the suppose gas ion half cell or the metal insoluble half cell was being taken. That means different electrodes were present in the different both the compartments, that was in the cathodic and the anodic compartment, right? But this EMF, it can also be produced when we are having the same type of the electrodes on both the ends, that is in the cathodic and the anodic department, but the concentration of the ions or the activity of the ions that are contained in that electrolytic solutions that are present in the cathodic and the anodic compartment, when they are different, then also this EMF can be produced. This is what is the case in the concentration cells. Okay, so let us see what more differences are going to arise in this case. So the first major one is this, that in both the compartments, that is in the cathodic and in the anodic compartment, we have the same type of the cells are present, same type of the half cells are present, but their concentration of the species that are present in the electrolytes, they are the different. This is the major difference in the concentration cell. Okay, so let us just write about this a bit. That is, this EMF, it can also be produced when the species present in both the compartments are same, but the activity, or we can call it as the concentration, because for the dilute solutions that gamma factor is equal to the unity, so the concentration is equal to the activity only. So the concentration or the activity or the concentration are different in both compartments, both half cells you can also call it as. Okay, so now let us take one of the example for this, then you can get a more clarity of this. So we are taking, suppose we have the half cells in both the compartments, that is in the cathodic and in the anodic compartment. Suppose metal ion half cells are present made up of the zinc. So we are taking the example of the metal ion half cells. Okay, so suppose here this is the anodic compartment where zinc is being oxidized, then comes the salt bridge, this gets reduced to the zinc solid. Here also it is solid and this is the aqueous phase. Now here in the anodic compartment, the concentration or the activities of the zinc ion, suppose it is A1, and for the cathodic compartment it is A2. Okay, so let us write the reactions for this. So in the anodic compartment we will be having the oxidation, that means the zinc changing to the zinc 2 positive plus the loss of 2 electrons. Okay, and in the cathodic compartment we have is the reduction, that is zinc 2 positive gaining 2 of the electrons and it is being converted to the zinc solid. Okay, but the activities, they are different. So in the anodic compartment the activity of the zinc 2 positive ions was A1 and in the cathodic compartment it was A2. So these 2 electrons in both of these get cancelled out in writing the overall reaction, and this zinc solid is present on both the ends. That means for the solids generally the activity or the concentration, it is taken to be the unity. So that means they both are not going to be involved in the overall cell reaction. So here it is going to be zinc 2 positive with the activity A2 going to the zinc 2 positive with the activity A1. This is going to be our overall cell reaction. Okay, so now according to the Nernst reaction, that was E cell equal to the E naught cell minus of the 0.0591 upon the N, that is 2, or we can write it here as N, log of product concentration upon the reactant concentration. So here we are seeing that in both the compartments, that is in the cathodic and the anodic compartment, same type of the cells are present and same type of the electrodes are being used. That means in the cathodic also zinc is present and in the anodic also zinc is present. So that means the E naught cell is going to be equal to 0, because the amount of this you can see that E naught cell, this E naught for the oxidation of the zinc to the zinc 2 positive, this we are to include, plus the E naught for the reduction, that is for the zinc 2 positive going to be equal to zinc 2 positive going to zinc. So we know that E naught for the oxidation, it is equal to the negative of the E naught for the reduction process, right? So here these both are the similar. Now the same species is getting being oxidized and the other one is getting being reduced. So that means these both are equal. This implies our E naught cell is equal to 0. So this is the another major difference, that is in the concentration cells the E naught cell value, it is equal to 0. Okay, so now the E cell, it is going to become, then E cell will be minus of the 0.0591 upon the N log of the product concentration upon the reactant. Here the zinc was present, so the reaction, in the reaction it was zinc with the activity A2 going to the zinc to the activity A1. So my E cell will be minus of 0.0591 upon the 2, because 2 electrons are involved here,